All right, welcome to the second part of Chapter 6, Reproduction at the Cellular Level. This time we're going to start by looking at binary fission in a prokaryotic cell, and then we're going to look into how mitosis can lead to cancer. First, in a prokaryotic cell, there's only one chromosome. So we don't need to have the big complex mitotic spindle. What happens instead is when it's time to reproduce, the chromosome migrates to the cell membrane. And in the cell membrane, enzymes in the cell membrane are going to copy the chromosome and make two complete new copies that are going to stay attached to the cell membrane. And then the cell is going to start building new cell wall and cell membrane between the two, physically pushing them apart. And when they're far enough apart, then the membrane is going to begin to pull in and grow together to form a septum. And when the septum is fully formed, now we've got a chromosome on each side, we've got cell membrane inside cell wall, and now the two cells can pinch apart to form two new daughter cells. So this is possible because there's only one chromosome. If there were multiple chromosomes, this would be a lot harder to do. So when does mitosis become pathological? Pathological means it leads to disease. And usually it's when you have either a problem with your checkpoint proteins, so that the cell just keeps jumping right into mitosis and skipping interphase with no resting period, or the mechanisms that would stop an abnormal cell and make it die, those stop working. And because these mutations are prior to cell division, they're going to be passed on to the new cells. And so these new cells can form a neoplasm, which is a growth. It's an accumulation of cells that have lost control about how they grow and divide, and they're not going to be able to differentiate into specific roles. They're basically just going to accumulate and have the potential to draw nutrients away from other cells and tissue, and that's when it becomes pathological. So genes for proteins that promote mitosis are proto-oncogenes. For the cell to jump into a new phase of the cell cycle, either to go from G1 to S, S to G2, or G2 to mitosis, we need proto-oncogenes to turn on and tell the cell that you need to move forward into the next stage. They're called proto-oncogenes because a mutation can turn them into straight-on oncogenes. Oncology is the study of cancer, and so an oncogene is any gene that can help cause a normal cell to become a tumor cell. Okay, and a neoplasm that forms a lump is a tumor. Okay, so this is showing you, these are cells that have not differentiated, and they're accumulating, and they're not going to be a normal cell, and it's just a lump. Okay, now. You also have proteins that can sense if a cell is damaged, called tumor suppressor proteins. And when they sense that there's a damaged cell, they can either cause the DNA to repair itself, or they can cause apoptosis to occur, which destroys the cell, programs cell death. Now, if mutated, then these tumor suppressor proteins don't stop a damaged cell from reproducing. They allow it to become a neoplasm. And now we've got a tumor. The examples of these are the BRCA genes that are associated with breast cancer. We already talked about P50. Well, we haven't talked about P50. P53, P21, RB1 are all proteins. Well, RB1 is a protein associated with looking for damage in the first part of mitosis. P53 and P21 are proteins specifically looking for damaged cells. There's a connection between viruses and cancer. We know the human papillomavirus can cause a cell to make proteins that interfere with its tumor suppressor proteins. So it's not able to take out a cell that's replicating out of control. Okay. So here's how P53 works. In a normal cell with normal P53, if you have some kind of damage to the DNA or some cell cycle abnormality, hypoxia means not enough oxygen, becomes anaerobic. P53 can arrest the cell cycle. If it can fix the DNA, it'll initiate DNA repair. If it can be fixed properly, then it picks up where it left off. If not, then it can program apoptosis, program cell death, and now this cell is not replicating anymore. It's gone, and it can't form a tumor. However, if p53 is mutated, it doesn't recognize that there's a problem with the DNA or a problem with the cell, and the cell cycle continues, and the cell survives. And now it survives. It can continue to divide and form other cells just like it, and now we can have a tumor. And if it's malignant, it can lead to cancer. Okay. Now, not all neoplasms are cancer. Okay. There are benign neoplasms that are just a growth, like a mole, or a skin tag, or some kinds. There's fascial ganglion neoplasms where it's just a lump of cells, but it's not really a problem yet. A malignant neoplasm is when it's a problem. This is when it's dangerous to health, because when you have a cancer and these cells are growing out of control, they end up crowding out other cells, tissue, and organs. And so this disrupts your physiology, your ability to function. Okay. And they can also break free and spread. A benign neoplasm, the cells don't break free and spread. They stay where they're at. So even if it was getting bigger, we could go in and remove it by surgery, or we could hit it with radiation and kill it all. But a malignant neoplasm, when cells break free, they can move and invade other tissues, and that's metastasis, and that's bad. Because in metastasis, here's our tumor, right? But now a cell breaks loose, it gets into a blood vessel, it moves to a distant part of the body, and now it can start another tumor. And when you hear them talk about, stage 1 versus stage 4 cancer, this is what they're referring to. Have they caught it before metastasis occurs, before it becomes metastatic? Because if you can catch it early, it's a lot easier to treat. Okay. I think a big part of fighting cancer in right now is early detection. When I was a kid growing up in the 1960s, if somebody got cancer, you started to say goodbye, because by the time they realized the person had cancer, there was nothing they could do about it. At that time, they just didn't have the therapies we have now. They didn't have the early detection, and so it had already advanced to a phase where it couldn't be treated anyway. And generally 3 to 6 months, and they were gone. Now we know that early detection is the key to fighting cancer. And so you're always, you're checking you go to a dermatologist and you have them check your skin and look for things like a basal cell carcinoma or a squamous cell carcinoma or a melanoma, which is the worst kind, right? And so you want to be checking yourself, especially as you get older. When I was in my early 20s, I had a housemate who had just had a skin cancer removed, and 3 or 4 years later, she ended up dying of skin cancer because it returned and they didn't get it all, and it spread to vital organs. So even skin cancer can be a real problem. But early detection is the key. Okay. 3 characteristics of cancer cells: they grow and divide abnormally. Okay. They're growing out of control. They're getting bigger. They're taking up space. They have abnormal plasma membranes, cytoskeletons. Their chromosome number can be radically different from normal chromosomes. Their metabolism is different. And they don't stay anchored properly because there's adhesion proteins that are missing or defective in their plasma membrane, so they can metastasize and move to different parts of the body. This is a karyotype of a cancer cells. This is a single cell from the HeLa cell line, Henrietta Lacks cell line. We'll talk about that in a minute. All right, there should be 2 copies of each chromosome, and there's not. There's 5 of number 1, there's 3 of 2, there's 3 of 3, 3 of 4, there's 7 of number 5, and some of them have been damaged. And the only one there's 2 of is number 22. Okay? So we can look at the karyotype and very quickly identify a cancer cell as being, abnormal in terms of the chromosome arrangement. The HeLa cell line, this is interesting. There's a book out, The Immortal Cells of Henrietta Lacks. Henrietta Lacks was an African-American woman in the Baltimore area who contracted and died of cervical cancer more than 50 years ago. In fact, it was really closer to 75 years ago now, is in the 1950s. And at the time, she was going to Johns Hopkins Hospital. Now, Johns Hopkins Hospital was built to serve the underprivileged population, people who were poor. who couldn't afford medical treatment. And so she was getting free medical treatment. And at the time, there was a researcher there who was collecting tissue from patients and trying to grow it in petri dishes. He was the beginnings of what we would call cell culture today. And every tissue sample that he took from someone, and they either donated to him or they collected as part of a surgical procedure, he was able to get it to go through a couple of cell divisions, a couple of mitotic divisions, but then they would die out until he took a sample from Henry Lack's cancer tumor. And he was able to keep growing it over and over and over. It became immortal. Immortal means they don't die. Okay? Why are cancer cells immortal? Well, we're going to learn later on when we talk about DNA that on the ends of your chromosomes there are repeating sequences of nucleotides called telomeres. And every time the cell divides, it loses some of the telomeres. And eventually, when that cell's divided enough times, the telomeres have shortened so much that the cell doesn't divide anymore. It slips into G0, that phase where the cell's not dead, but it's never going to replicate again until it finally dies or is removed. Okay. Well, cancer cells have the enzyme telomerase. They can fix their telomeres, and that's why they're immortal. Okay. Now, Henrietta Lacks died, but they were able to keep her cells alive all this time. And in fact, there are many different HeLa cell lines they've developed. They've been sent all over the world to research facilities everywhere. We've learned a tremendous amount about viral growth and protein synthesis and the effect of radiation and how cancer functions by using these cells. Okay, now she didn't give her consent for this. She was dead. She died by the time they figured this out. Okay, her family hasn't gotten anything from this. Billions and billions and billions of dollars have been generated from the knowledge that has been learned by using the HeLa cell line. Okay, we've used the HeLa cells to test Taxol, which is a drug that helps to interfere with mitosis of cancer cells and to kill them. Okay, today you have to sign a consent form before anybody can have any of your tissue. And you say, well, why was this allowed? Well, remember, she's getting free health care in the 1950s. And the researcher who did this with her cells, he didn't make any money from it. In fact, he would give those cells away to anybody who wanted to work with them. He'd pack them up into a special container and fly them all over the world. Okay, so, what happened then couldn't happen now. But we learned a tremendous amount from Henrietta Lacks cells. And we have to give, that's not something she intentionally intended, but that's why she's famous. Interestingly, the book, The Immortal Life of Henrietta Lacks, the proceeds from that go to the family to get them educational scholarships and health care so that they're finally able to get something beneficial out of it. So that's the end of part 2. Next time we're going to talk about meiosis. Start chapter 7 and talk about meiosis and gamete formation.